Hi everyone, so I'm going to cover the bacterial genetics lecture, and in this lecture we're going to talk about a few things. We're going to talk about how you get diversity between organisms, specifically how we have so much diversity between bacteria. We're going to learn about mutations and different types of mutations, and then finally we're going to end the lecture by talking about different types of gene transfer. So we're going to start out by what the word mutation means. A mutation is any change in the DNA sequence, specifically the DNA nucleotide sequence. So when we look at a DNA nucleotide sequence, remember that DNA can be made up of 4 base pairs: adenine, guanine, cytosine, and thymine, which we abbreviate. So when you change any one of these bases, that is what we call a mutation. We're going to talk about the different changes you could do to the DNA nucleotide sequence, but it's basically changing that sequence where we get a mutation. And mutations, typically people think of them as bad things, but I want to state that mutations, they can be negative. They cannot cause anything, and sometimes in rare occurrences, they can be positive. They can create something that no one ever knew. So there's a lot of different results of mutations, and we will talk about those. And mutations can occur in 2 different ways. You either have spontaneous mutation, which from the name, they occur spontaneously from completely random errors that happen during DNA replication, or mutations can be induced by mutagens. And those mutagens can be physical or chemical agents. And we're going to discuss both. Right now, we're going to talk about this one, spontaneous mutations. So with spontaneous mutations, when we look at a gene, and remember a gene is a DNA sequence, you can get spontaneous mutations, and the type of mutations you can get are nucleotide substitutions, which we also call base substitutions, because you're specifically focusing on the nitrogen bases, which are the A, G, T, C, or you can get nucleotide deletions or insertions. So just be aware that sometimes people will just say base substitution, nucleotide substitution. So base and nucleotide can be interchangeable here. For substitution mutations, they can lead to 3 different things, and we're going to talk about them. So if you substitute one DNA base for another, for example, if you substitute like an adenine to a guanine, as a mistake that happens, it can lead to 3 different effects. So base substitutions can lead to silent mutations, missense mutations, and nonsense mutations. And we're going to discuss what each of these things mean. Another type of mutation that can happen is when you actually completely delete a base or you insert a base. And when you do that, that can lead to what we call frameshift mutations, and I will discuss that. So here are the type of mutations and the effect. And again, the type of mutation and the effect it has. Okay, so now just specifically focusing on base substitutions, or again, what we call nucleotide substitutions. I want to mention that sometimes these are called point mutations because you're affecting one DNA base pair or one DNA nucleotide. And remember, with base substitutions, when you switch one base for a different base, you can get 3 different outcomes. You can get a silent mutation, you can get a missense mutation, or you can get a nonsense mutation. And we will talk about this. So for point mutations, it's a change in just one DNA base, and it results either in missense mutations. So remember that after we do transcription during protein synthesis, once you have your RNA molecule, the 3 base pairs are in what we call codons, and you are going to translate those codons to amino acids. So we're going to talk about the effect that has. And remember that when we look at the genetic code, every codon codes for an amino acid. So if you do a base substitution and you changed one base for the other, and it changed the amino acid from one amino acid to a completely different amino acid, we call that a missense mutation. If you actually change the amino acid to a stop codon, we call that a nonsense mutation. And if the change from one DNA base to a different DNA base resulted in the same amino acid, we call that a silent mutation because nothing happened. So for example, when we look at the codon AAA, it codes for lysine. AAG also codes for lysine. So if you were to have that mutation where an A accidentally got mutated to a G, you still get lysine, so the effect is it's still a silent mutation. And when we look at this figure, this is a good review of transcription and translation for everyone. So here's the DNA template strand. And remember, you transcribe DNA to messenger RNA. So you can see the DNA here was transcribed to messenger RNA. And that's done through RNA polymerase. Now we have our mRNA strand, which has the 3-letter codons, and we're going to translate that to amino acids. So AUG, for example, if you look at the genetic code, it codes for the amino acid methionine. AAG codes for lysine, etc. So let's imagine that this gene here coded for lactase, so the lactase enzyme. So let's assume that these bacteria have the lactase enzyme and this codes for that. And enzymes are proteins, and proteins are made up of amino acids. So let's assume that this is what lactase is supposed to look like. So this is what we have if things are going correct. Now let's say a mutation happened and T accidentally got changed. And here, instead of having what you would have had here, which was C, cytosine, it got changed to T. Because remember, a mutation is a change in the DNA sequence. But when you change the DNA sequence, it affects everything else. So it affects transcription and translation. So the complementary to T is A, and now the codon you have is AGC. In the original strand, GGC coded for glycine. Now AGC codes for serine, a different amino acid. So we call this a missense mutation because one amino acid got changed to a different amino acid. Now, if we had a different mutation, where if we look at the original strand, here we were supposed to have a T and it accidentally got mutated to an A, it randomly did, then this led to a stop codon. This is called a nonsense mutation. And if the mutation led to the same amino acid, then we would call that a silent mutation. And just to talk about major effects that one base pair can have, the disease sickle cell anemia. So sickle cell anemia is a disease that we see. So normally red blood cells are nice and round. When people have sickle cell anemia, their red blood cells are what we call sickled. So they're kind of curved. And because of that, they don't hold as much oxygen as they should. So sickle cell anemia is a disease, and it's caused by a point mutation that changes one base pair to a different base pair, and unfortunately this leads to a completely different amino acid, so that leads to a missense mutation. And here's an example of that. So on normal circumstances for red blood cells, there's a lot of molecules involved in making red blood cells, but one of the proteins is hemoglobin. And in hemoglobin, this is the sequence that you're supposed to have. But with people with sickle cell anemia, A got mutated to T, and then when you transcribe that, you get a different mRNA, and then when you translate that to amino acids, you get a different amino acid. So this leads to very different looking cell types, and it leads to the disease. So point mutations can result in different amino acids, which again are called missense mutations, and this can ultimately lead to a protein that's completely non-functional. Maybe sometimes it's slightly functional. So they can lead to a lot of things. Now we're going to talk about a different type of spontaneous mutation. So before we talked about base substitutions, now we're going to talk about what happens when you add or delete bases or nucleotides. So as you can imagine, this can lead to detrimental effects because you're really having a big effect. And the effect we call is a frame shift mutation. So Is the addition or deletion of one nucleotide usually most of the time has a pretty bad effect. And that's because imagine when you add a nucleotide, you shift the whole RNA codon. That's why we say it leads to a frame shift mutation. And if you delete one of the nucleotides or bases, that also leads to the same thing. So you can see that instead of altering one amino acid, which you potentially do with nucleotide substitutions, here you're changing the whole amino acid sequence. So that's why we say it leads to a frame shift mutation. And usually they're detrimental, meaning they are bad. So as a review of mutations, the effects of different types of spontaneous mutations. With spontaneous mutations, you can have nucleotide substitutions or nucleotide additions or deletions. With nucleotide substitutions, it leads to three outcomes. You can get a silent mutation, you can get a missense mutation, and you can get a nonsense mutation. With adding or deleting bases, it leads to a frame shift mutation. So we were talking about spontaneous mutations, and now we are going to talk about what happens when you actually induce mutations. So induced mutations are induced through physical or chemical means, and there are different environmental agents that cause DNA changes, because remember that a mutation is a change in the DNA sequence. So for physical mutagens, we have UV light, sunshine. So sunshine is a mutagen. That's why sunshine can also cause skin cancer, too much sunshine. And another type of radiation, physical mutagen, is ionizing radiation. And this is why you hear that getting x-rays all the time is not good, gamma rays. So these physical mutations cause thymine dimers. And a thymine dimer means that on a DNA strand, because that's where we see thymine, thymine will bind to thymine and it'll almost cause a break in the DNA. And so it causes a lot of issues going forward because the thymines become cross-linked and it's hard to do any more replication, any more transcription, translation. So this is what radiation causes. And this is a type of physical mutagen. Another type of induced mutations can be caused by chemicals, and different chemicals will cause different types of mutations. So there's base pair mutagens, meaning that they change base pairs, so different chemicals can change pairing. We also have base analogs. There's chemicals that we use in medicine that mimic what actual DNA nucleotides look like. Now in medicine, this can be a good thing to mutate a defective gene or something that's defective, but it can also have harmful effects. And we have chemicals that cause frame shift mutagens. So there's many different chemicals that can cause mutations. One chemical that everyone has heard of is nicotine, which we see in smoking. So the reason why nicotine is bad is because it can mutate DNA. Another type of chemical mutagens is some chemicals are base analogs and they mimic bases. So there is a chemical molecule called AZT and it looks very similar to thymine. And the problem with this is that when you give AZT to cells, because it looks so similar to thymine, they'll take it in. And once they take it in, this can affect replication. So one of the first drugs to be used with HIV treatment was AZT. And so here we talked about, again, how we can induce mutations. These are not spontaneous, and you can induce mutations through radiation or through different chemicals. Now, mutations are happening all the time, and the question is, how do your cells fix them? So repair of mutations, there's a lot of different ways, but we're only going to focus on two for purposes of this class right now. So how do you repair damaged or mutated DNA? There is mismatch repair, and we're going to talk about another one in a little bit. So let me go back. So with mismatch repair, it repairs point mutations and it removes mismatched bases and all the other bases that are around that base that got changed. And what happens is DNA polymerase is always proofreading because DNA polymerase is involved in replication. And when it makes a mistake, it'll actually go back and do mismatch repair. And once you cut out all the bases that shouldn't be there, use DNA ligase to join the bases. There should be an image here. I don't know why it's not showing up, but you should have it in your PowerPoint. Another type of mutation is thymine dimers. And remember, this is when thymine binds to another thymine. This can be repaired by nucleotide excision repair. And these enzymes cut out incorrect bases, and then DNA polymerase comes and fills them in. Okay. We're going to talk about a different topic right now. So we talked about mutations. Now we're going to talk about gene transfer. And this is how organisms, bacteria specifically, transfer genes between each other. We're going to talk about how gene transfer is used in technology and medicine. So just a few definitions to begin with. I want everyone to know genetic recombination, and this term is the exchange of genes between two DNA molecules to form new combinations of genes. So this is how we create diversity in a population, and the organisms that are created from this change of DNA molecules are called recombinants. And the cell that actually gives a portion of its DNA to the other cell is called the donor cell. The cell that receives the DNA is called the recipient cell. And when the donor's DNA is put into the recipient's DNA, the recipient is now called a recombinant cell because it has a cell with donor DNA in it. So it's a combination of DNA. That's why we call it recombinant cell. It has a combination of its own DNA and another organism's DNA. So one way that we get recombinant DNA is the human insulin gene that comes from humans. We incorporate it in bacteria's DNA. And once this human insulin gene is put into bacteria's DNA, we have a recombinant cell because this cell has a combination of human DNA and bacterial DNA, DNA from two organisms. And once you put that DNA back in the bacteria, this is called a recombinant bacterial cell. And this is very useful. We use recombinant bacteria to make insulin for us because bacteria are dividing all the time. So you can get bacteria to make you a lot of insulin and then extract the insulin protein and make it available as medicine for people who need it, specifically for people with diabetes. Gene transfer, some more terms. There's two types of gene transfer. There's something called vertical gene transfer and horizontal gene transfer. Vertical gene transfer is when you transfer genes from parents to offspring or organism to its offspring. And vertical gene transfer happens in plants, animals, all organisms. Bacteria, when they divide, if you want to call their division their offspring, they're transferring their genes to their offspring. So when animals, humans have children, that's vertical gene transfer. So it's just a transfer of genes from an organism to its offspring. Horizontal gene transfer is something very unique, and it is only done in bacteria, and it's the transfer of genes between species. So it's when one bacterial species can transfer genes to another bacterial species. And we do not see this in humans. I gave this example in class, but this is an example if, for example, someone had a different eye color than myself, and I was able to touch hands with them and get that eye color. That's a transfer of genes, which we humans obviously don't have. But this is why we see so many different types of bacteria, because they can do horizontal gene transfer. So specifically, bacteria can transfer either pieces of their DNA to other organisms or whole plasmids. So with segments of DNA, this has a narrow host range, meaning that it only happens between very closely related species. Something else that bacteria can do is bacteria have plasmids. And when we talked about it in class, we said that plasmids are extra chromosomal DNA, extra DNA. They're these round circular pieces of DNA that hold non-essential genes, such as antibiotic resistance, toxin production. So bacteria can transfer whole plasmids to other bacterial species. And with whole plasmid transfer, it can be done either between closely related species or many unrelated species. And again, here is horizontal gene transfer. So this organism has a gene for antibiotic resistance, and it was able to horizontally transfer it to another bacteria. And the original bacteria that had this, for example, is E. coli. And here it gave that gene, it transferred it to Streptococcus pneumoniae. So this is why we see bacteria developing so much antibiotic resistance. One reason. Plasmids, as a review of what plasmids are, again, they're pieces of DNA and they can replicate themselves. They have the means to replicate them. They're extra DNA, extra genes. They're much smaller than the actual bacterial chromosome. The bacterial chromosome is what actually holds the crucial genes that bacteria need. Any plasmids that bacteria have are just extra DNA. And plasmids usually hold DNA that code for proteins that make a bacteria more pathogenic. Remember, pathogenic means things that make a bacteria cause disease more. So if something is pathogenic, it causes disease. And some bacteria, many bacteria actually can transfer plasmids between each other. Methods of gene transfer. So now we're going to talk about, well, how do bacteria, how are they actually able to do horizontal gene transfer? How do you transfer genes from one species of bacteria to another species of bacteria? And there's 3 different methods of how gene transfer is done. So bacteria can transfer genes between each other through transformation, transduction, or through conjugation. And we're going to talk about each one of these things. So transformation is when bacteria can take up DNA from the environment. So we could say naked DNA transfer. It's basically if bacteria are in the environment and there's pieces of DNA, if they're able to do transformation, they're able to take it up. So some bacteria have this ability, others don't. Transduction. Transduction is a very unique way where bacteria get different genes through something called a bacteriophage, and we're going to learn about those later, but what bacteriophages are, what a phage means virus, and a bacteriophage is a virus that infects bacteria. So in transduction, a bacteriophage, again, which is a virus that infects bacteria, will infect bacteria and insert its genetic material. So that's how bacteria will get some new genes, another form of gene transfer. And conjugation is when bacteria can contact each other through pili, we will talk about that, and transfer genes between each other. So these are the 3 different ways that you can get gene transfer: transformation, transduction, or conjugation. So let's focus on transformation. So transformation is cells taking up naked DNA. When we say naked DNA, we mean DNA that's just found in the environment. It's not in a cell. So basically, if you were to do this artificially in lab, you have the bacterial cells and you can just pipette on them whatever DNA you want them to take up. And if they do have the ability to do transformation, they will take it up. And only one DNA strand enters, because remember, DNA is double-stranded. And then after the DNA enters the bacterial cell, you actually need to get that DNA into the bacteria's own DNA. So something called homologous recombination happens. And homologous recombination is when that foreign DNA is incorporated into the host bacteria's DNA. And now we have a bacteria that's been transformed. So that was transformation. Transduction. So transduction is, again, when bacteria get new DNA using bacteriophages. And bacteriophages are bacterial viruses. And then once those bacteriophages put their DNA, it gets put into the own bacteria's DNA, the host's DNA. So let me show you guys what happens here. So Here you have a bacteriophage, and it will infect this bacterial cell. It'll inject its DNA in the bacterial cell, and the bacteria will take this piece of DNA, and through homologous recombination, it will get put into their own DNA. So we're going to learn all about viruses later, but this is one method where viruses can change cells. Finally, conjugation. So conjugation is when bacteria can just exchange DNA between each other using pili. Remember we talked about during cell structures, we said that some bacteria have pili, and pili are like these hair-like appendages on bacteria if they have them, and one function of pili is to do conjugation. So if this bacteria has pili and this bacteria has pili, the pili, and they're in close contact, the pili will connect and the bacteria can exchange DNA between each other, and you have to be physically connected with each other close. You can't have like one bacteria here and one bacteria a mile away and they do conjugation. They have to be very close to each other. And then DNA is exchanged between cells by pili. You can exchange either pieces of DNA or a whole plasmid can be inserted. And I didn't really mention this, but when bacteria share their DNA, it's more like sharing because you don't lose the DNA. You give them a copy. So conjugation plasmid transfer. So when this picture is not showing up, but if you guys download my lecture on the Canvas website, you have the lecture, you have the pictures. So what happens is we call the cell that gives up its DNA the donor cell. And in science, we say that's the F+ cell. F stands for fertility, so it's going to give up a piece of its DNA. The recipient cell is called the F- cell. And remember that both donor and recipients get plasmid copies. Please look at this image in your actual lecture when you download it. Another way that DNA segments can be transferred. So before we were talking about plasmids, we're going to learn about something called transposons. Transposons are jumping genes, and they're small DNA segments that can move from one piece of DNA to another piece of DNA. Transposons are very unique. They really are jumping genes. So after a plasmid is transferred from one bacteria to another using conjugation, using the pili, if that plasmid carries a transposon, different genes can jump into the actual recipient cell chromosome or DNA. So just to sum up, I apologize. I don't know why these images are not showing on here, but again, you have the images. So we talked about different types of horizontal gene transfer, and gene transfer and mutations is why we have diversity. So diversity, the way you get diversity between organisms is through mutations or gene transfers. And remember, mutations can happen spontaneously during replication, or they can be induced. And although many mutations are thought to be harmful, they can be beneficial because they can bring adaptations to an organism. They can add a lot of diversity. So we talked about this in class. This is a microbial class, but for example, eye color, eyes that are not brown are thought to be as a mutation. So there was a deletion that happened where people lost the ability to make melanin, and that was not a harmful mutation, so it was maintained in society. Another type of mutation that could happen is maybe bacteria mutate and all of a sudden they can make a toxin, and maybe that toxin kills host cells, host human cells, and so they're able to survive better. And then gene transfer, and we talked about 3 methods of horizontal gene transfer: transformation, which is naked DNA transfer; transduction, which uses a bacteriophage; and conjugation, which uses pili. And natural selection is the survival of the fittest. So why are some people still born with sickle cell anemia? So there's a big question in evolutionary biology for why do we still have mutation? And as you guys can imagine, sickle cell anemia, we can talk about it later, but sickle cell anemia protects you from malaria. So with malaria, it's easier to get if you have your round, healthy red blood cells, and it's harder to contract if you have sickle cell anemia. So this is why sickle cell anemia has still been maintained in society. Blue eyes have been maintained because they're not harmful, and so they weren't a harmful mutation. And then bacteria that are antibiotic resistant, as you can imagine, it can be very beneficial for a bacteria to not be harmed by antibiotics. And these are all things that were caused by mutations. And many, many more diverse organisms are diverse due to different mutations that they've gained over time. So bacteria maintain their diversity again through mutations and gene transfer, and there's so many species of bacteria because of this. I'm going to end this lecture talking about MRSA. So MRSA is methicillin-resistant Staphylococcus aureus. So methicillin is an antibiotic, and Staphylococcus aureus is a type of bacteria, and this bacteria was not resistant to antibiotics to begin with, but over time it gained the gene to be resistant to antibiotics, and most of the antibiotics in the psyllin antibiotic family, and this was done through mutation, and this mutation benefited the bacteria, and this is why also we see so many antibiotic resistant organisms. So we are going to end this lecture. So this is a summary of everything we've been talking about. And here are study questions for you guys, practice tests, and you have your lecture to look at. Okay, so go ahead and look at your lecture, and hopefully everything makes sense.